This is the immune system part 4. This is the last part of the adaptive immune system and talking about humoral immunity. So we've talked about cell-mediated immunity, and now we're going to move on to the very last part. So the adaptive immunity was divided into cell-mediated immunity and humoral immunity, and the T cells were our cell-mediated immunity, and then our plasma B cells are going to be our humoral immunity, which makes the antibodies. This is the very, very last stage of fighting an infection. Selected B cells produce copious amounts of antibodies that interact with antigens that are found on the pathogens, and they need the support of helper T cells. And if we remember, helper T cells specifically are going to address cells that are infected when they release granzymes and perforins to kill those cells, but they don't create antibodies. An antigen is a material that stimulates selected B cells to produce copious amounts of antibodies. So when we talk about the idea of a pathogen versus an antigen, the pathogen is the infecting material itself. The antigen is the label on that will create the response in the B cells. Antigens include bacterial cell walls, viral parts, flagella. It is not the actual bacteria, but it is the cell wall of that. An epitope is the part of the antigen in which the antibody attaches, and so B cells make these antibodies, and then the antibodies attach to the antigen. An antigen can have more than one epitope or type of epitope, and so it can have multiple areas that the antibody can attach to. These are some examples of different epitopes that are found on two types of antigens, which are on pathogens. One pathogen is a protein, one is a bacteria, and then on its surface it has antigens, and the antigens have epitopes on them. How does humoral immunity work? So naive B cells that are not predisposed to do anything yet, they have receptors that are different for each B cell. When the antigens attach to the receptor site of the B cell, the antigens are brought into the cytoplasm of the B cell. The antigens attached to the MHC2 proteins are transported to the cell membrane. So the antigens are internalized, and then they are attached to the MHC2 proteins, the superficial proteins. An activator T cell, or a helper T cell, which has been activated, makes a match between the MHC2 protein, which has the antigen presented on it, and the AG complex. The helper T cells secrete something called cytokines, which causes the B cell to reproduce and make plasma B cells and memory B cells, all of which are eventually going to make antibodies. So, for example, for this selected B cell, thousands of B plasma cells are cloned. They're identical to the B cell. They're just called B plasma cells as opposed to B cells in general. The plasma B cell can synthesize over 2,000 identical antibodies per second, which are the things that are going to attach to the antigen and help kill it. And we see it looks like this. This illustrates the thousands of different naive B cells. Only the one with the correct receptor site is selected and cloned. Otherwise, we will be selecting for B cells that will not create the correct antibody. There's multiple ways that antibodies can inactivate antigens. These are a couple examples. There are basically three that will enhance phagocytosis or lead to phagocytosis. The three that lead to phagocytosis from a macrophage, if we think back to the very beginning of immunity, neutralization, where a virus or a bacteria is basically coated with the antibody. Agglutination, basically the antibody is going to bind to the antigen and pull them into a large group, so they are in a large cluster, more easily consumed by the macrophage. And the precipitation of soluble antigens, basically the clustering of antigens that are soluble using a certain liquid, which then can in turn be engulfed by the macrophage. The last one leads to specific cell lysis as opposed to a cell eating the other cell. It's called the complement fixation. Basically what happens is the antibodies start to make holes in the foreign cell or the antigen. Eventually that leads to cell lysis, or the explosion of the cell. Again, just a different image of the thing that we were talking about before. When we start talking more about how we inactivate an antigen, opsonization, certain bacterial cells have so many Ag determinants that the antibodies coat the bacterial cell. The constant regions stick out. We call them the Fc regions, and the phagocytes have Fc receptors, allowing the phagocyte to roll over the pathogen, and phagocytosis occurs. Essentially, this allows for, this is how, if we look back, how neutralization and agglutination occur, right? This idea that the antibodies are going to basically coat it. They're going to have certain regions that are recognizable by the phagocytes, the Fc regions, and then between that interaction, we're going to see phagocytosis occurring. This is just another image of how that is happening. Feel free to pause and look at this if you'd like. There are videos on YouTube. Please look them up. It will help quite a bit. So what happens when we need to stop this response? Because our body doesn't need this response all the time. What is going to happen is we're going to see regulatory T cells helping to halt the immune response. We don't really know how they are activated. People think that potentially antigens do the activation. They're not quite sure. It's thought to stop any B and T cells that don't have a purpose yet from being activated to having a certain purpose. Then the immune response eventually stops, and the activated cells, the ones that are still producing, will die because of their short lifespan. The only cells that are going to stick around are the memory cells. And this is kind of a really good summary image of humoral and cell-mediated together. Feel free to pause and look through this. Memory B and T cells are selected for when exposure to subsequent pathogens occur. Basically, the next time I'm exposed to a certain disease, my memory cells, there we go. My memory cells are going to quickly reproduce, make more plasma B cells, helper T cells, and cytotoxic T cells to attack whatever pathogen they've already been exposed to before. And so the life cycle of a memory cell is actually very long because they need to stick around for a very long time, whereas our normal B and T cells die off after the response is no longer needed. Plasma B cells begin to make AB antibodies, and cytotoxic C cells begin to destroy infected cells. So again, once my memory cells make the respective T cells and B cells, they will start a response immediately. It is why when we are exposed to a pathogen the second time around, it is very rare that we get sick. This is a really good image of that. Feel free to pause and summarize this in your notes. So when we talk about artificial immunity or preventing diseases with vaccines, what we figured out was that subsequent exposure to pathogens doesn't result in the disease. So the first time I will get sick, my body mounts an immune response, I get better. The second time, my body is already ready, I have memory B and T cells, and so my active immunity causes those cells to be made, and the pre-made antibodies kill off anything without my immune system having to kind of get running again. What this means is that I rarely will get sick again when I'm exposed to the same disease. If we go back, the idea behind immunity or vaccines is that we are injecting people with a form of the disease that will not make them sick, but will make the body create memory cells and antibodies. Once those memory cells and the antibodies are present, they will not get sick again. So when I traveled to Africa, I had to get the yellow fever vaccine. The yellow fever vaccine is actually the live yellow fever virus, which I did get a little bit sick when I took it, but it made it so that my body has those memory cells. It has yellow fever antibodies in my body, so if I were ever exposed to it in Africa again, I don't know if I was, I wouldn't get sick. Typically, they try to avoid injecting the active disease because that can actually make you sick. They've found ways to neutralize it while still getting the same response. There's a couple types of immunities in regards to if we're getting it passed on from something else. Natural activity immunity. Organisms are exposed to the actual pathogen and get the disease, and then we don't get it the next time. This is very normal. Think chickenpox. You are now immune to it if you've had it. I know that they have a vaccine now, but think back to when I was a kid, we actually got chickenpox, and then the next time when we were exposed to it, we wouldn't get it. Artificial active immunity, this is vaccines. We are given an inactive part of the pathogen, so my memory cells are made, so the next time I'm exposed to it, I don't get the disease. And then natural passive immunity, there are other ways that we can get antibodies. The most specific one is through breast milk and through the shared placenta with the fetus. This can last a few weeks to a few months, which is because there are no memory cells that are passed on, just the antibodies. So this is a really good protective measure for infants, but eventually it does wear off and children have to form their own immunity. When we talk about artificial passive immunity, right, or this idea of, we talked about the other three types, patients exposed to a serious pathogen can be given a shot with preformed antibodies against the disease. So in our other passive immunity, right, we talked about how I actually get that passive immunity on my own. They have done research to try to figure out how, when people are exposed to a serious pathogen, if I just give them the antibodies, not the memory cells, but the antibodies, could we potentially prevent them from getting the disease? The way that we would harvest antibodies is we would find the plasma of a person who's had the disease and basically extract the antibodies from the blood and inject it into someone who has been exposed to the disease. This is kind of up and coming. The idea is that if I'm exposed to, let's say we talk about bird flu or something like that, or Zika, which is a virus, so it's a little bit different. But the idea behind it would be I would, if I knew for sure that I was exposed to a disease like this, instead of going through the first round of getting sick, being injected with someone else's antibodies would automatically get me to the second stage of the immune response, meaning I would not get sick. There's a lot of issues with this theory, but it's an idea that people have. We've talked a little bit about vaccines. We're going to move on for now. What happens when the innate immune system goes awry? Certain bacterial infections can induce an overwhelming inflammatory response. Too many mast cells, too many phagocytes, huge amounts of cytokines and histamines are released. Blood pressure drops dangerously low and tissues can swell a lot. This is when we have like very serious physical trauma to the body. The body is being inflamed in so many places that it cannot help itself. Other reasons when the immune system can go awry, not all self-reactive lymphocytes are destroyed in development. Those that are survived are suppressed. Another thing we talk about is autoimmune diseases, where certain suppressed lymphocytes are activated and they attack our own body tissue, which can be a really big or large issue. We're going to talk about blood typing in class. I'm just going to flip through it quickly, so you're welcome to pause the PowerPoint if you'd like to, but we are going to talk about this in class.